I'm going to show you how to do a Plasmid mini prep. You will be given directions that look just like this, and everything we need will be given to you in Eppendorf tubes. But here's our culture. This is our culture of P. mycomar. So it's an E. coli that has P. mycomar, the plasmid, in it, as we discussed in class. These are different buffers that we're going to use for the actual Plasmid prep. It's very easy, and so those will all be set up for you. You'll need a pipetteman, and then you'll need tips. You'll need a microfuge. You'll need an Eppendorf tube to pellet your cells, and then this is the magical stuff. This is what comes with the kit. This is a little glass-filled, impregnated little mini Eppendorf tube, essentially. But this is where you're going to catch your DNA, and you elute it from this white stuff, and we'll talk about that. And then this is just a collection device. Okay, so that's essentially an anion, or not anion. It's an exchange column, essentially, and we'll show you how that works. So, first things first. When you read the directions, it says centrifuge bacterial cells, 15 to 20 seconds at high speed. Okay, so what you need to do is take out, I would take out a full milliliter myself of your cells and spin them. Take out your little cells, carefully pipette them out, put them in an Eppendorf tube. And because I'm doing this by myself, I'm going to do this twice because I want to have a balance. And you can do the same if you'd like to balance your tubes. Well, not like to, you have to. So I'm doing this twice. Okay. Got our little cells. So we're just going to come over here, pop it in the centrifuge. Make sure you put them on opposite sides, like usual. Put your top back on, however that fits. Close this. And in my case, I want this to be going for, it says, blah, 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 15 to 20 seconds at high speed. So we're going to go all the way to high speed. And we're going to go and count off 30 seconds. And this recipe is very easy to do. This is what comes with the kit. Anyone can do it. It works really well. And right now, all you're doing is spinning your cells out to get them into a pellet. Very simple to do. That's probably enough. So we're going to turn off. And these centrifuges, just like the big one, they won't open until it comes down. You hear it click. There you go. And you can see that there's a little pellet in there. Okay? Now you gotta remove the supernatant. Because this is pelleted pretty strong, I'm just gonna take this and just shake it out into a beaker. You can very carefully remove that too if you're really, if you're worried about it. But that pellet is pretty tight in there. All right. So it says now 200 microliters of P1. So you set your thing here. This is basically an alkaline lysis protocol, by the way. Very simple, something that everyone uses. Very efficient, works really well. Here's our P1 buffer. They're color coded, which is nice. So it says 200 microliters and resuspend completely. So we take our 200 microliters. We resuspend completely by repeated pipetting up and down, just like we did before. There she is. Completed. Okay. Now it says add 200 microliters of the green buffer and mix by inverting 2 to 4 times. So what you're doing is prepping this for lysis, and now you're going to lyse it. Lyse the cells and get all that goodness out into. So I'm going to put that in. And right now, they're being lysed. And all their DNA and things is being loosened, pulled out from the body of the little bacteria into the solution. And you can tell this is mixed because when I open this top, you should see a stringy, see that stringy stuff there? That is nucleic acid, more than likely. You see that little gooby string? Okay, so that means they're lysed. Okay. Now it says add 400 of P3 buffer, which is the yellow buffer, which is right here. And it says do not vortex. Okay, mix gently but thoroughly. That means invert it a few times. Okay, so we'll go to 400 microliters. And this is the only step these things have to be, or this yellow buffer has to be refrigerated or stored in refrigeration. We'll take our 400, pop it in there, and it says, you put it in here, and what you do is you mix it by gentle inversion, and you'll see that all this white flocculent stuff starts to show up. So what you're doing is renaturing all of the DNA, and you're supposed to do this until the pink is gone. Okay, so it's basically there's a pH indicator in there, and so you keep inverting this back and forth until all the pink is gone. And again, what you're doing is you are now, you lysed the cells previously, you got all the DNA, the plasmid and the nucleic, the genome out, okay, the chromosome, and you have, you denatured it, so it all pulled apart, but now you're renaturing it really quickly. And that renaturation process is such that the genomic DNA is so big it won't renature, it turns into all this flocculent garbage. But the little plasmids are small enough and they will renature and come back together into a little circle. Okay? So now it says incubate at room temperature 1 to 2 minutes. So we'll do that. Now, it says here centrifuge for 2 minutes, and that means at full tilt. So come back over here, balance everything. And we turn this thing on high speed for 2 minutes and let it go. Okay, she's coming down after our 2-minute run. Now when you pull this out, you'll notice that you've got a bunch of this supernatant and you got that white flocculent pellet. Okay, the next step, you're going to take all this clear stuff which has our plasmid in it. This down here is all the nucleic acid other than the plasmid. It's fats, it's protein, it's all kinds of junk you don't want. Okay, so we're going to transfer this super into our little column that we just simply assemble by grabbing this column piece and putting it in the collector. And all you're going to do is put the contents of that yellow stuff into this little column. And the only watchword here is that depending sort of on your organism, this can be a tricky step just because you don't want to pick up any goobery stuff. You want to just get the liquid up here. So you start pulling up liquid, and as you get to the bottom, things get, I'm just putting it right in that little other tube, things can get a little dicey on you. Because as you pick this up, I hope this works. I lucked out. I had no problems picking up, I didn't pick up anything extra, which is great. But ordinarily you do. So, here this is this little sample thing. Pick it up, put it in this little thing, so it's in this column. Alright, now it says, take this in this little container, just the way we have it, and it says blah, 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 blah. centrifuge the column for 30 seconds. Again, this is at high speed. So you put this in here. It looks like it's not going to fit, but it will. And there's no top on it. That's okay. 30 seconds. See, in the next step, what we're doing is we're flowing through everything through the column. And what's happening is all the hopefully plasmid is getting stuck on that white stuff that's in the base of that column because of charges. Because now the rest of this is basically washing. It says discard the flow through, because that's what you don't want. Your plasma will be stuck to that white particulate stuff at the bottom of that little column. Okay, so I removed this after a 30-second spin, and it says discard this stuff. Because now this stuff is stuff we don't want. This has got all the extra stuff. Our plasmid should be stuck on that little white stuff. So we discard this. I keep it around because I'm going to use that little column later. But it says here, add 200 of Endo wash buffer. So Endo wash buffer is basically washing your little plasmid, getting rid of various things you don't want in there. So I stick this back in there again. Take my Endo wash buffer. Maybe shake it up a little bit. I don't know how long this has been sitting. You can see it's got detergent in it. And lots of salt and stuff, I think. 200 of this. And what it says is, it says 200 and spin the column. So when you're adding this, you just put it in here and just drop it right in over the column. Just like that. No fuss, no muss, nothing exciting. Don't pipette up and down, just drop it in there. Now we're going to spin this, and then it'll be pulling that endo wash through that white thing, and you're washing our plasmid essentially. So we do the same old thing. Stick it in, 30 seconds, let it go. So we've lysed our cells, we've we've denatured the DNA, we renatured it, we've spun out and gotten rid of all the GAC and nucleic acid, the big stuff, the genome, and now we're washing our pellet. We stuck our pellet, our little plasmid, to that white piece in the bottom of the tube, and now we're washing it, okay? Now it says 400 of plasmid wash buffer, so that's another buffer we're going to wash and centrifuge for a minute. So it's 400 microliters this time. That's 30 seconds about. This is harder to do and show you guys how to do it than it is for you to do it. It's very simple. Nobody will mess up, okay? So now it says 400 of plasmid wash buffer. Take our plasmid wash buffer. I'll have all these out in Leppendorf tubes so that it's easier to do. So it says add it, just like we did before. Okay. But now it says spin it for a full minute. So now, after this, the plasmid will still be stuck to that white stuff. Okay, it's still stuck there, but it's washed and clean. Now what we'll do, it says, is transfer the column into a clean centrifuge tube. That's what you're going to collect in. And how do you collect? Well, you're going to hit it with a small volume of what's called DNA elution buffer. This could be water, this could be something called TE buffer. Basically what this is, right now there's a lot of salt, and the DNA is stuck under those conditions to that white silica stuff in the bottom. But now if we suddenly add a bunch of very low ionic strength stuff, i.e. something that's basically water, now the plasmid will fall off of that matrix and it will come through. Now we want to save what comes through, okay? So we're going to put a very small amount, 30 microliters is a good amount. I've got my littler pipette here, set at 30. Kind of get it ready. So there we go. So now we can toss that. We'll stick this into our new tube. And now we're going to add a low ionic strength buffer to this, 30 microliters of it. And when you put this on here, just make sure you don't touch the matrix. Never touch the matrix, but you just plop it right on top. There it goes. Nothing magic again. You can't close the top. You just leave it like this and it will spin just fine, okay? It seems sacrilegious, but it'll be good. I tend to put it over on the side like that. So the top is kind of leaning over a bit. Switch top back on. And it says blah, 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 30 seconds. And now what we'll have is our washed and clean plasmid down in our little collection. And now we'll take it over and, well, you will take it over and find out how much DNA you have and how pure it is by putting it into something called the nano drop. Okay? And that's very easy to use. We'll just use that when we do our thing in class. Okay? I won't show you that on video. I put that little face on there when I bought this centrifuge in 1998. It's still there. Now, you've got, this is your column that now you're done with. Toss it. And now you're left with 30 microliters of plasmid, we hope. So what you're going to do, like I said, is after that step, you're going to grab me and I'll show you how to take this over to Dr. Stenbeck's and Dr. Zanis's lab, and we'll figure out how much you have and how pure it is. That'll let us know what volume of this to use when we electroporate our mixos, which is our next video, is going to be how to get the mixos ready to be electroporated, which means making them competent, and how to do the electroporation itself.